Hi, it's my pleasure to introduce Professor Ho Leung NG, who will be talking about open source COVID-19 drug discovery. Professor Ng is an associate professor of biochemistry at Kansas State University in the USA. Professor Ng's research is in computational and experimental drug design for cancer, immunology, and infectious disease, especially using artificial intelligence. Dr. Ng's training is in biochemistry. He was an undergraduate at Harvard University. Dr. Ng completed his PhD at the University of California at Los Angeles and was a postdoctoral fellow at the University of California at Berkeley. Dr. Ng is the founder of Open Source COVID-19, a nonprofit group dedicated to COVID-19 drug discovery and open science. Dr. Ng has a strong interest in global scientific issues and cooperation. Take it away.
Okay, great. Let me... All right. Can everyone see the screen? Okay, great. So thank you for the very kind introduction. So today I want to talk to you about some of our work on open source COVID-19 drug discovery. So so my basic background and research is primarily in the area of biophysics and biochemistry as applied to drug design. One thing that's unique or special about our group is that we do both computational and experimental research in our own lab, which is nice because that allows us to experimentally validate our computational work. So for a long time, I've been interested in open source approach to problem solving inspired by the success of Linux. So Linus Torvalds, the founder of Linux, he started Linux as a hobby project when he was a college student. So here is his first public post on the Usenet news groups saying, hey, I'm doing a free operating system. It won't be big and professional. This has been brewing and he's starting to get ready and he's looking for people to help him out. And since that initial post, the Linux operating system has gone to take over essentially the entire world. And now pretty much every server, every supercomputing platform now runs on the Linux operating system. This operating system was built free. The source code is freely available, and it was at least in the beginning was primarily contributions from volunteers. So can we replicate this kind of success in drug discovery, especially with the goal in mind of producing drugs in a way that is less expensive, perhaps more creatively, and again with the goal of having drugs that can be not just work, but truly globally available to people around the world. And so this is an especially important for infectious diseases like malaria, tuberculosis, and now COVID-19. So one of the leading drug discovery platforms, organizations in this field is Open Source Malaria. So I've been working with Open Source Malaria for several years. This group is led by Professor Matt Todd from University College London. And to summarize the goals and rules of open source malaria, the guiding principles are that it's based on openness, transparency, non-ownership, so that all work is in the open and is released for public use, no patents, inclusion, so anyone can participate. Matt or anyone else is not in charge, and that discussion will be is public, so that anyone can freely discuss and criticize and give feedback. And we think that this kind of open science should lead to more higher quality and more reproducible work. So based on my experience working with open source malaria, early on in the pandemic, I started open source COVID-19. So like OSM, we're a nonprofit open source group where our data and products are freely available to the public. It is inclusive, meaning that anyone can join our consortium as long as they're a scientist. Right now, we have more than 20 global members and partners from mostly academia and also some former pharma people. And through this network, we have extensive assets that range from computational work to bench work in the way of chemical synthesis, biochemical testing, to crystallography and testing with live virus. So our current targets are the coronavirus M-Pro protease. So this is a protease that cleaves the viral polypeptide that allows it to replicate. And then the now famous spike protein, which is the exterior protein of the virus responsible for recognizing the human receptor. So most of our work has been focused on M-Pro protease. From the beginning, this was recognized as the most promising small molecule inhibitor target. And most viruses have some kind of protease, and they are proven drug targets and are the targets for clinically used drugs against HIV and hepatitis, among others. So the proteases differ quite a bit from virus to virus. And so with the case of the SARS-CoV-2, this is a protease that is somewhat conserved across coronaviruses. So there's also this long-term hope that we could make a pan-coronavirus drug. And what we see, what we're looking at here is the recognition site for the peptide cleavage site. And so what we see here is that at certain sites, some amino acids are highly conserved. So that gives us some hope that we could make some kind of peptide or peptidomimetic that binds in this site that cannot be cleaved and would jam up the works of the protease, making it unable to replicate. Like a lot of groups who worked in the very beginnings of the pandemic, our first strategy was to do repurposing. So most of you by now have heard quite a bit about repurposing. Repurposing is an important strategy because it's the fastest way to get us to a usable drug, or at least a usable drug hit. So what repurposing involves is using, ideally, trying to find a clinically used drug, so it's already been FDA approved, and hopefully finding that it also works against SARS-CoV-2. And so the way we did this is that we screened the drug bank database. So the drug bank includes all FDA-approved drugs, as well as drugs that have moved to, that have gone into clinical trials or have been described in the literature as being advanced preclinical stage. The one thing that we did that was different from other groups is that we also looked at the adjacent chemical space of these drug bank molecules, meaning that we looked at all molecules that were closely related to the drug bank drug. So this would expand our database by an order about tenfold. This was done by virtual screening. And so virtual screening, of course, is quite fast, and we were able to screen the non-covalent inhibitors, non-covalent drugs, inhibitor drugs of drug bank quite quickly, and really and then test them experimentally against an assay with the purified recombinant M-Pro protease. And we did not find anything that was especially promising. So what we did, and what a lot of other groups did not do, is to perform virtual screening of covalent inhibitors. So because there's a covalent reaction involved, this requires a different kind of virtual screening that is much slower, not nearly as automated, requires a lot of manual definition of potential ways that different kinds of atoms and could react and the different kind of bonds that would make, and then what would be the products. These usually had to be specified to the software. So it's quite a bit of work. But this is important because a lot of the known inhibitors of viral proteases do work in a covalent manner. So we did this, and we also identified some promising hits. And again, we sent them for validation by our partner at the Worcester Institute in Philadelphia. And then our third strategy, which I'll get into some detail later on, is using AI methods to generate completely new inhibitors. So by far our strongest hit from the computational screen for covalent inhibitors was this molecule called GC376. So this is an experimental veterinary antiviral originally developed for and validated against coronaviruses in cats. So those of you with cats probably have heard of this, but coronavirus infections in cats is a somewhat common and unfortunately very deadly disease. So this was shown to be effective in a small scale clinical trial for cats, and but it had not yet been tested in humans. And so we found that this molecule, and then several other groups at about the same time, also identified this same molecule as being quite promising for the COVID-19 virus. So from the beginning, there were several liabilities to this molecule that we recognize and were described in the literature. And so, let's see. Let me try to get a laser pointer. So there we go. So here, there are several. One is that the initial covalent warhead is quite reactive. And so from talking to the former pharma scientists in our consortium, they strongly recommended going with the weakest covalent warhead possible, because this would make it less likely to have off-target effects and toxicity. So this is especially important for COVID-19, which in the large majority of cases leads to fairly mild symptoms. So we don't want to make the medicine worse than the disease, of course. Now, if you were to decrease the reactivity of the warhead, your drug, while being less toxic, will also likely be less potent. So you need to make up for that by increasing the binding affinity of the drug. So we found two places where one could make potential substitutions to increase that potency. And then finally, the original drug was only had rather poor pharmacokinetic properties and was not orally bioavailable. So this is often the case with peptide drugs. They're still too peptide-like, and they go into the body and they basically get chewed up by your digestive system. So ideally, we would want to make this drug, modify this molecule to make it less peptide-like and make it something that one could take as a pill instead of requiring IV administration. So our first generation of compounds substituted first a nitrile group as the covalent warhead. This is one of the mildest commonly used warheads in drugs. And then a less common azonitrile group. So this is interesting. It's it is fairly reactive, but at least in practice, it has shown to be with some other compounds, it has shown to potentially be less toxic. And so we are finishing up the synthesis of these two compounds and should have them ready for testing within a matter of a month or so. Now, about half a year, about 6 months after we started our project, Pfizer announced that they also were working on this and that they had some compounds. So last year, they announced their first compound, Pfizer PF 07304-814. And this is also a peptidomimetic that actually is you can see here, very closely related to GC376, and that they were planning to enroll patients in phase 1 clinical trials. So that felt very good to us, because that I felt that we were on the right track. We're an open source group. We're not competing with Pfizer or anyone else. In fact, we hope that that when we get our data and our experiments all completed and out for sharing to the public, that Pfizer or any other group can use our results to improve their research and improve their drugs. And then earlier this year, just about 2 months ago, Pfizer announced a new inhibitor, PF 07321332. This was a big deal because now you see this is orally available. And what they did is they still kept quite a number of the same elements of GC376. Now you also see that they've switched to the nitrile warhead that we are experimenting with. And then they tacked on some parts from a hepatitis C drug, boceprevir, to make this orally available. Boceprevir itself is not effective against the coronavirus, but they're able to use the chemistry for this part of boceprevir that made boceprevir orally available, and then transfer that onto their anti-coronavirus drugs. So I thought that was quite smart. And so hopefully what we plan to do is that we'll also start work on our second generation of antiviral drugs that will be orally available. Okay, so now I'm going to switch gears a little bit and talk about our efforts using artificial intelligence methods for drug discovery. So there are two main ways that AI is currently used for drug discovery. The first is primarily in terms of predicting drug activity properties. So this would include docking, virtual screening, QSAR, quantitative structure-activity relationships, and then I left out predicting PK, PD, and toxicology properties. And then finally, at a little bit more bleeding edge is what we call generative molecular design, using the AI to come up with drugs without a predefined chemical library. So what this has been described as an inverse design problem. So one specifies certain functional properties that you would like your molecules to have, and then you let the AI algorithm explore the chemical space and then come up with molecules for you that fulfill those desired properties. So many of you, of course, are familiar with this kind of generative AI, where AI has been trained on millions of photographs on what it means for a picture to be human, of a picture to be that of a human face, without explicitly being taught that this is a human face. And the power of these kind of AI has really increased a lot, to the point where now all the faces shown here are AI generated. And at least at a very quick glance, unless you examine it very closely, at least to me, they all look very real, like they are real people. So what we are trying to do is take this same kind of approach and say, can we train AI using thousands or maybe millions of molecules and have AI come up with new molecules? We train it with drug molecules. Can AI come up with new drug molecules just based on being able to identify what makes drugs, certain molecules, drug-like? And then there are very many ways by which one can define the molecule, ranging from familiar methods like fingerprints and smile strings to using defining them in terms of atoms and bonds, to more physical chemical methods, defining them as electron density. So the representation of the data is important. So this is an especially important goal, because chemical space is virtually infinite. And the space that we've used so far for drugs is a very small sliver of that large chemical space. Maybe we're missing out on all kinds of potential drug molecules because we just haven't explored that chemical space yet. This is work that is led by Professor Alana Spuruguzik at the University of Toronto, our close collaborator. And so this is a problem because just like a lot of other fields of science, medicinal chemistry, drug chemistry is based on what we've done in the past. What we've done in the past has been closely might be a very narrow part, a small part of chemical space. And it turns out, let's say we have a completely new disease and a completely new drug target like we have here. Well, a viral drug target is not a viral protease is not completely new, but still there aren't that many viral proteases in our knowledge base compared to, let's say, GPCRs, G protein coupled receptors. That single family GPCRs represents roughly something like 40% of all known drugs. So our a priori knowledge and state library of drugs that are used for compounds used for high throughput screening will be dominated by GPCR molecules. So this is a technical schematic. I don't want to get into it too much for this audience, but basically the approach we have been using is combining a genetic algorithm with a deep learning discriminator neural network. So the genetic algorithm generates a molecule, and then a discriminator is a neural network that has been trained to evaluate whether that molecule is good or not. And then this works in an iterative process. And so this is what it looks, this is the kind of results that we are getting at this point. So we start with just putting a methane molecule near the binding site of the drug 3D crystal structure. The algorithm evolves this methane molecule and it grows in complexity over time. Already you can see that by generation 25, it has evolved some rather large, complex molecules. But you look a little more closely, you can see that the molecules have rather weird chemistry, nothing that one would consider drug-like. This is a limitation that we recognize and we're working on. We have made quite some improvements, really through trial and error. So this represented the kind of molecules we had in 2020, and by this year, now we have molecules that look like this, still far from perfect, but we're getting something that is a little bit more drug-like. But nevertheless, even if these molecules that are the products of the AI cannot be directly used or cannot even be synthesized, they provide a very useful starting lead for a chemist. A chemist can look at this and look at the proposed binding and can easily come up with ways to transform this molecule into something more practical. So here, for example, we look at, in blue, GC373, and this is from a real crystal structure. So we know its binding mode, and here is the AI-generated molecule, and we see that there is quite a lot of overlap. But the AI molecule explores some areas and some chemistry not seen in GC373. So that suggests to us, hey, maybe, for example, the AI molecule moves into this binding pocket, and GC373 has not gone up here. Maybe the potency of GC373 could be improved by accessing this binding pocket. Another obvious idea that comes out of this is that, hey, perhaps we can combine these two molecules and make some kind of macrocycle. And then that would be something that no one has done yet, as far as I know. And that's pretty cool. Okay, and finally, just want to talk to you about some of our very preliminary work on trying to target the spike protein with small molecule inhibitors. So with the spike protein, we started with, as far as we know, as I know, there are no small molecule inhibitors that have been described for spike. Spike is more like a receptor. It is not an enzyme, so it's not the matter of just coming up with an enzymatic inhibitor. So one can come up with a peptide that comes from the ACE2 receptor that spike binds to. But as I mentioned before, peptides and close peptidomimetics often do not make for good drugs because of poor PK/PD properties. But what we can do, so what we did is we used this the generative AI algorithm, and we also use our own algorithm for identifying potential small molecule binding sites. We found a potential small molecule binding site on the spike surface here. It's a relatively small site, so that will be a challenge. I'm not sure if this can really be done. And then what we did is we used the generative AI to come up to map this surface, and then we use the results of that to then to use the pharmacophores identified by the generative AI to perform a traditional virtual screening of a database. And so here we have a molecule that we've identified that we have not tested experimentally yet that could potentially fit into this space, although the binding is relatively poor, but it's a start. So I don't know if this will lead anywhere, but what we will do is we can hopefully experimentally validate some of these results, and maybe some other groups can build upon this and make a stronger and more useful spike inhibitor. So to summarize, we've done quite a bit in terms of using computational methods and then validating them experimentally. What we need to do, of course, is improve the drug lead likeness of the AI results. We need to improve the synthetic accessibility of the AI results. There's no point generating molecules that can't be synthesized in a practical manner. One way of doing this is to incorporate fragments in the molecule building process. And what we want to do, of course, is to improve the oral bioavailability of our protease inhibitors. So I want to thank the people who've worked on this. So for my own group at Kansas State University, led by these 5 postdoc students and graduate students and even undergraduates. OSC 19, a large consortium, but special people who have participated, who have really made important contributions, including three very experienced former pharma scientists who've been giving us practical advice on drug discovery. Our collaborator Troy Messick at the Worcester Institute, who leads the biochemical studies, and then Alana Spuruguzic's group, who leads the AI development. Our early work was sponsored by the NSF. We've been generously funded by Intel and NVIDIA, who have been interested in the AI and computational aspects of our work. We hope to be able to get NIH or some other source of funding to move this faster. Thank you very much for your attention. I look forward to answering any questions.
Wonderful. Thank you so much, Professor Ho Leung, for your insightful presentation. The floor is open for any questions or remarks for Professor Ho Leung from the Kansas State University.
I thought I wanted to jump in here. This is on Dr. Andrea. Congrats on a really innovative program, and I'm just blown away. I have a quick question for you with respect to the path to the clinic. I know that certainly in the COVID area, there are a lot of programs that were designed to accelerate the path from discovery to the clinic. Are you able to tap into those? And are you looking to do your clinical work on your own, or is that something that you're going to be partnering to get to the clinic?
Right. So great question. So in terms of the work that have been done to accelerate, at least on the preclinical stage, they really haven't gone that much further than what we did. And we did this with almost no budget. So that wasn't as promising as advertised. But that's something you don't know until you try it. So in terms of the next stage of accelerating the drug discovery, we can do some of the work. We have an expert who does viral studies with mice, so we can do that within our consortium. But essentially we will need more help in the future. So our goal is not to go from A to Z and present a drug to people, to the patient. We will take this as far as we can. Right now we have almost a minimal budget, and we'll look for partners who are willing to do this and move this forward as we get closer and closer to moving this to the clinic.
Oh, excellent job.
Wonderful. All right. Again, if anybody has any other questions for Dr. Ho Leung, you can always email us and we can forward it to him as well. And I do want to thank you again for your amazing presentation, Professor.
Thank you.
And with that said now, we will be heading over to our first networking break. So it will be until 11:05. So if you haven't had a chance to have breakfast, this is a good time to do so. Feel free to also pop in and out of the networking rooms, the booth, to collect the passcodes to redeem for virtual prizes. We have a lot of great prizes like Amazon gift cards, Uber Eats, and Apple Watches raffles that will be happening. So feel free to pop in and out. You guys have till 11:05, so we'll be on break till then. Thank you very much.